Hello marine biology students. This week we're going to be talking about invertebrates in the ocean. These are animals without a skull and a backbone. And the first that we'll be talking about is the simplest, the sponges. So we'll be discussing many invertebrate phyla this week in lecture. So what is an invertebrate? Well, animals without a backbone are known as invertebrates. Whereas if an animal does have a skull and a backbone, then it is a vertebrate. Turns out about 97% of all identified animal species fall into this category of invertebrates. They are by far the most abundant and diverse categories of animals. All major animal groups, or phyla, have marine representatives, meaning individuals who live in the marine environment. There are no phyla which are exclusively terrestrial. However, there are several phyla that are exclusively marine, meaning they only live in the oceans. So let's look at what it takes for something to be considered an animal, and then we'll talk about the different groups of animals. For something to be an animal, it needs to be eukaryotic, multicellular, and often with tissue-level organization, although as we'll see with one animal group, they lack that tissue-level organization. Animals are heterotrophs, meaning they aren't photosynthetic. They have to feed on other organisms. They cannot feed themselves with sunlight. Animals require oxygen. For aerobic respiration, because they are multicellular, they often have high energy demands. They need to have oxygen present for the breakdown of food for energy. And lastly, most are mobile, at least during some portion of their life, meaning they can move, although in certain organisms in their adult stage they will be sessile or non-moving. So when we talk about animals, we need to discuss how their body is organized. And it turns out there's a few different symmetry plans when it comes to basic body characteristics. One is radial symmetry. With radial symmetry, there's clearly a top and a bottom of the organism, but there isn't clearly a right or a left side. You could almost imagine a terracotta flower pot, which is open on one end and closed on the other, and you can rotate it 360 degrees, and you aren't going to clearly see that one side is different from the other side. That is radial symmetry. Conversely, bilateral symmetry. Bilateral symmetry is when we have clear top and bottom, and it can be split down the middle, and right half and left half will be symmetrical to each other, but they will be distinct. They will be mirror images of each other. And then lastly, organisms that lack tissue organization often lack body plan design or symmetry, and so we call these asymmetrical. So asymmetrical animals, they don't have a defined, distinct body plan. And those will be the ones we're looking in this first group, the sponges. So here we can see examples of radial symmetry and bilateral symmetry. With radial symmetry, again, we can see that it doesn't matter which point you divide it from that center point, the two halves are going to look the same. Whereas with bilateral symmetry, there's a clear front and back, top and bottom, left and right, and it's symmetrical along one plane of similarity. So looking at our major invertebrate phyla, we have the poriferans, which are the sponges, which we'll be talking about in this video, The cnidarians and the ctenophores, which we'll talk about in our next video, those are going to be things like jellyfish and coral and comb jellies. Then we have some bilaterally symmetrical worms, and we'll talk about those in one group, but they are phylum Platyhelminthes, which are flatworms, the Nemertians, which are ribbon worms, nematodes in phylum Nematoda, arrowworms in phylum Chetognatha, and then also our segmented worms in phylum Annelida. The next group that we're going to look at are the mollusks in phylum Mollusca, arthropods in phylum Arthropoda, and then a group of organisms that have a similar feeding structure called a lophophore. These are our lophophorates in the bryozoans, the pheronids, and the brachiopods. And then we'll finish things off with our discussion of the echinoderms, and then the hemichordates and the chordates. So quite a list of invertebrate phyla, although believe it or not, this is probably only about half of the phyla in the animal kingdom. So sponges, Phylum Porifera. The basic characteristics of the sponges is that they are sessile as adults. So they're attached, they don't move as adults. There are numerous tiny pores on the outside of the body known as ostia, which allow water to flow in through them. And that's really how the sponge is going to be getting its nutrients, by filter feeding water as it comes into its body. The water flow will also carry waste and gametes away. Sponges are primarily found in the marine environment. As I had mentioned, they have an asymmetrical body plan. Meaning that there's no defined symmetry to their body shape. When we look at the types of cells that make up sponges, there are a few different types, although they are not organized into tissues. So the first cell type we'll talk about are the collar cells, also called choanocytes. These choanocytes, they have flagella and a collar of cilia on one end of the cell. These choanocytes line the interior canals of the sponge. And the flagella they have create water currents that causes the circulation of water through the sponge. This circulation of water brings food in from outside, and also again carries wastes and gametes out. So it's the collar on the choanocytes that end up capturing the food particles that are brought in by the water current. Now another type of cell that are in the sponges are going to be the pinacocytes. And these are flattened cells that cover the exterior of the body, almost like a layer of skin or a layer of protection. Another type of cell in the sponges are the porocytes. And these porocytes can regulate the amount of water entering the sponge at different areas. When we look at this diagram of sponges, we can see the general vase-like structure with ostia leading in, and then the osculum being the major outcurrent of the water. And then as we zoom in and look at the side, we can see the pinacocytes making up the boundary, the porocytes controlling the pores, and the choanocytes that are in the interior chamber. Now another type of cell we haven't quite talked about yet are the amoebocyte cells, and they can wander around the inside of the sponge. They have a variety of purposes, but some of the important ones include generating spicules and also spongin. When we look at the structural support of a sponge cell, it's full of this elastic protein fiber known as spongin. And that's where some of its structural integrity comes from. Some sponges will also have spicules. And these spicules can either be made out of silica or a calcium carbonate. Spicules come in a variety of shapes, from simple rods to star shapes. In fact, there are some that look exactly like the star of the Mercedes-Benz symbol. Now, both of these structures, the spongin and the spicules, are secreted by internal wandering cells called amoebocytes. And these amoebocytes can actually also become the other type of sponge cells as well. When it comes to reproduction, sponges are able to reproduce both asexually and sexually. Asexual budding is one form of reproduction in which a larger adult starts just forming smaller individuals that separate and are able to live on their own. They break off and they grow into a new sponge. Experiments were done to separate the cells of a sponge by pushing that sponge through a sieve or through some screen mesh. And in that way, the sponge cells were separated from each other. But in a petri dish, those sponge cells would end up finding neighboring cells, and they would end up forming structures together, forming new individuals. Now, a surprising thing was that if multiple sponges of different species had their cells separated in this way and were set to grow in a petri dish, the cells of the different species would actually separate and segregate from each other. So you would end up with small little clusters of sponge cells of different types. So there's some form of recognition between the cells as to what species they belong to. So experiments with artificial reproduction by fragmentation showed, again, that when those sponges were pushed through sieves, they will reform into smaller individuals, and that different species of sponges seem to recognize their own cells. Sponges also reproduce sexually. This sexual reproduction of sponges involves sperm cells of the sponges being brought in by the water currents and finding an egg that is found within the interior of that sponge. Fertilization takes place, developing into an embryo and then a larva. That larva is then released from the excurrent siphon, and it can travel a certain distance from the parent and then settle into its own adult sponge. So while adult sponges are not motile, the larva can travel a certain distance before forming a new sponge. Most sponges are hermaphrodites. Meaning they can produce both male and female gametes. So that finishes our introduction to the sponges. Now before our next video, I would like you to think about what it would be like not to have a right side and a left side. We'll talk about that in the next video.